All right, welcome everyone to this week's Oxford Neurotheory Forum. We're very lucky to have Dr. Nico Schuck here with us today. Nico is leader of the Max Planck research group Neurocode at the Max Planck Institute for Human Development in Berlin. And he's going to tell us today about distributed replay in the human brain and how to find it. And just before launching into that, I want to say a few words about Crowdcast if you're just joining us the first time. So first of all, you can ask questions as we go with this Ask Question button. So I'll be watching that, and if there's a clarification question, I'll pose it to Nico during the talk. And then if you have questions you think other audience members might be able to help with, you can also post that in the chat. And I won't monitor that as closely. And then we'll have questions at the end where if you like, you can come up on screen. So if you do submit a question that you want to ask at the end, if you could just add, do you want to be called up on screen or not? All right, and with that, welcome, Nico. Thanks a lot for being here, and over to you.
Thank you, Andrew, for the introduction and for having me, and for thanks everyone for coming and attending my talk. As you just heard, I'm interested in replay and in particular how to measure it in the human brain using fMRI. So I'll be focusing on some of the methodological advances that we have made in the past. Before I get started, here's a very brief recap. I'm assuming most people will know what replay is, but in the off chance that someone is not, I like to show this little video over here. So you will see a bright spot that is a lamp that's mounted to the head of a rodent, a rat. And the rat is walking around in a little maze that you can see. And what you can see on the left side of this video is recording the firing of cells from the hippocampus of that rat as it's navigating around, and they're like each row is one cell. So we can start watching the video, and you will see the rat starts moving around, and as it's moving around, you see that the cells recorded in the hippocampus in a very systematic fashion relate to the position of the animal in the maze. So the blue cells, which were like number 1 here in this example, fired when the animal was around here, and the yellow cells fired when the animal was in the top part of the maze, and so forth. So that is not new. What's interesting is the thing that happens next. So now if you pay close attention while the animal is standing. Yep. And there it was. So what you could see is that the cells, when the animal stopped walking around, the cells kind of went quiet for the most part. But then there was this reactivation event where a bunch of those cells were reactivated, as you can see by that stripe here. And what's interesting about that is, I guess at first, that now the play cells are activating, even though the animal is not in those locations that they originally were thought to signal. So they are, in a sense, offline, meaning that the animal is not actively behaving or being in these places, but they still sort of signal some mental process that refers to these locations. The other aspect is that it was really fast, much faster than the sequence of play cells that you had seen during the behaving part of the experiment. And the cells activate sequentially, which effectively just reflects the structure of the environment. So locations that are nearby in space, the corresponding cells were activated in sequence, as you can see here by that stripe going from the more bluish colors to the red color, which corresponds to the path the animal has taken. Just because it's so nice, we can watch it one more time. Cells go quiet, animal is standing, and then blop, there's another of these sequence events. Okay, so all of that is pretty well known, and following the initial discovery of that phenomenon, people have realized there's a pretty large diversity of replay phenomena, and replay actually comes in many shapes and sizes. So after having it discovered in sleep, they have found that it occurs also during wakeful rest periods, as you have just seen in this video. They found that it can involve previously visited paths, like you saw in the video, but it can also sometimes signal novel paths the animal has never taken before. When the animal is about to make a choice, the replayed sequences can either indicate a path the animal is about to take, that happens, but sometimes the replay reflect paths that the animal is not about to take, or particularly those that it's not about to take. Replay can be forward and it can be backward sometimes, and it's affected by a number of task parameters, such as whether an event was rewarding or not. And in addition to that kind of diversity of the features of replay, researchers have also noted that there's an anatomical diversity of replay in the sense that when recording outside of the hippocampus, in addition to the hippocampus, they found similar events in other areas. So people have found co-occurring replay events in visual cortex, in auditory cortex, in several regions within prefrontal cortex, in entorhinal cortex, in the ventral striatum, and that list probably goes on and will continue to grow in the near future as people begin recording in different places. So one idea that I find particularly interesting is that these two levels of diversity might be related. And I think that beautifully illustrated by a study from Freya Olafsdottir, who showed that replay in the hippocampus can be coordinated with replay of grid cells in the entorhinal cortex, but it is not always coordinated. And the distinction between when it's coordinated with the entorhinal cortex and when not is actually reflected whether the task, whether the animal is engaged in the task or not. And presumably that reflects a functional distinction between replay having a role in consolidation and when it's coordinated with the entorhinal cortex versus replay having a role in planning. And during these periods, replay may not be coordinated with the entorhinal cortex. So that suggests that by understanding the patterns and the diversity of coordination, we might also sort of understand the landscapes of different functions that replay could play in neural computations more generally. And things can also get quite complicated. So another interesting example is a study from Kiefer et al. From this year, in which they found fast reactivation events of cells in the medial prefrontal cortex. And they found that these reactivation events, first of all, reflected a different aspect of the task. The play cells in the hippocampus were reflecting the position of the animal. The MPFC cells were reflecting the distance to the goal, so more like a 1D location. And in their hands, the hippocampus replay and the MPFC replay were not, were not coordinated at all. So at the same time, the MPFC replay wasn't a chance event, but they showed that the replay in MPFC related to a specific cognitive function of the task, the rule switching in their example. So this is another example where understanding the diversity of replay events, where they occur in the brain, how they are coordinated, might give us further insights into the functions of replay. So that in combination kind of leads me to think that spatially resolved whole brain imaging will be a really useful tool to study replay. But the particular challenge that we have is that we need to measure really fast sequences while at the same time measuring them all over the brain with some sort of spatial resolution. And the other aspect that I like to point out is that it might also be useful to study that outside of rodents. As you have seen, all the examples I've made so far have been based on rodent recordings. And I think if we can make some progress of understanding the same process in humans, for instance, that's a species I find interesting, then that would be nice. So past research has, of course, already started doing that, using whole brain recording techniques to study replay in humans. And particularly noteworthy, I guess, here, speaking to an Oxford crowd, is the progress with MEG, and there are a number of really nice papers from Yancey Liu and Elliot Wimmer and Kurt Nelson and so forth. But one of the tricky parts of that is that MEG offers less good spatial resolution, and it seems particularly hard to disentangle between co-occurring but anatomically distinct replay events and understanding where they are, which kinds of information they reflect. fMRI, on the other hand, has also been used to study replay, but fMRI is slow, so it seemed unfeasible to use functional magnetic resonance imaging to really understand the fast sequential aspect of those events. And so in the past, most researchers have resorted to focusing on stable reactivation patterns during rest. That might also reflect replay, but they don't give insight into the sequential nature of the replay event. So last year, then, Yael, Nif, and I reported that after participants had done a decision-making task, they were resting in the scanner, and what we observed is that in the hippocampus, patterns that were activated during the task were reactivated during rest, and that wasn't new in and of itself, but we found that sequences of those patterns were reactivated in a manner that could only be reflected or explained by the sequences of task experiences participants had made immediately before. And that sort of gave us the first indication that tapping into the sequentiality aspect with fMRI might be feasible after all. So to be kind of a little bit more precise why that is surprising and interesting is because in fMRI, as I guess most of you will be aware, we are measuring neural activity very indirectly. And what we are measuring specifically is a delayed and sustained hemodynamic response that occurs a few seconds after neural events have actually occurred and lasts for a number of seconds. So as you can see here in this plot, when a neural event occurs around second 0, you will see that it will take a few seconds for the signal to kind of lift off at all, and then it will kind of lift off, but that will take sort of on the order of several seconds for it to go down again. So that basic property of the fMRI signal might make you think that it's unfeasible to use fMRI to study replay at all, because as you've seen in the video, replay is a really fast event that happens on the order of milliseconds. So the question that we now asked is, how fast can the sequences that we measure with fMRI, for instance, in this pattern sequentiality metric that we have observed, be in general, and what are the good ways to do that? So one kind of very, very basic argument that Yael and I had already made in the 2019 paper was that there is a very broad feature that gives us some hope about that signal. And that is, if you imagine a very simplified and kind of idealized scenario that you had 5 different place cells, they reflect 5 different locations, and they fire one after each other with a few milliseconds in between them. And we had 5 different voxels in fMRI that respond only to these 5 different places. Again, this is kind of an idealized example. What you would get is, of course, these highly overlapping curves, but you would see that the ordering and the strength of activation of every voxel at every point in time will reflect the sequence of the original firing as it has occurred. And that ordering signal means there is some information retained in the signal about the fast sequence, and that information is actually sticking around for quite some time, for the entire duration of the HRF. So for several seconds, we get to see information about, or get a chance to measure something about the ordering and so forth. Okay, so what we wanted to do now is to create an experimental task in which we could really test that. And for testing that, the key thing that we wanted to establish is that we have ground truth about fast neural event sequences. So we're not going to be talking about replay because it's spontaneous and we don't know for sure whether something occurred or not. We infer that from the data, but we're talking about an artificial setup in which we experimentally induced fast visual activations, and then we ask, can we decode the content of those fast visual activations? And the general setup is that we have a slow task on which we train the classifier, and then we take the classifier and apply that to the fast events. Okay, so the slow task is pretty standard. We use 5 different image categories. that you can see here from a classic paper from Haxby, and participants are seeing these different image categories. There is a sort of really standard fMRI longish intertrial interval, which gives us time to measure the associated hemodynamic response function after people have seen a cat versus a house and so forth. But importantly, what we intersperse with these slow trials are sequence trials in which participants initially see a verbal cue about one of the 5 categories, and then they are exposed to a sequence of all 5 categories, and that's the speed of that sequence vary from trial to trial. And once the sequence is finished, there is a longer delay, and then they have to answer a question about the initial cue, which is which position within the sequence was the shoe that we had sort of primed you before to watch out for. And as I mentioned, the sequences could be relatively slow with 2 seconds between each image to relatively fast with only about 32 milliseconds between each image. And to kind of give you a sense how fast that was, here is a little GIF of the 64 millisecond condition, so the second fastest one. And you can see it's indeed quite fast. I should also say it's not that the images are on the screen only for 32 milliseconds. The images are on the screen for 100 milliseconds, but then there is a 32 millisecond blank before the next one comes on. Okay, so classifier training was done on a slow task, and that's pretty standard. We show the 5 different image categories. We took a mask of inferior occipital temporal cortex that has been shown by a classic paper from Haxby to be sensitive to these different image categories. We use a 1 versus rest logistic regression classifier, and to kind of give you a sense of what's happening. Here are sort of the average standardized patterns from one participant within the mask in response to the 5 different image categories. And you can see that they're reasonably intermixed. All of them sort of elicit responses in these areas. And then the precise pattern is what the classifier learns to distinguish. So that works well. That's not surprising. We get a mean accuracy of about 69% of overall decoding accuracy. And when we take that classifier and now we apply it to every single TR following the stimulus onset, then we see that the classifier sort of traces the slow hemodynamic response function and is a bit more symmetric and maybe simpler than the original HRF, because now it's sort of a classifier that sort of extracts information from thousands of voxels rather than reflecting an individual HRF. So here you see the black lines are the probabilities of the logistic classifier of the true class. Gray lines are the not true classes, and you can see for each one of them, and each line is one participant, we get very clear signaling that the probability of a cat being shown sort of increases at about 40 hours after stimulus onset. So what we then did first is that we started modeling this time course of a single event, and we took a particularly odd model, and you will see later why we made that choice. Namely, we said we will just fit that as a snippet of a sine wave. So the sine wave, the full thing, is the thing that you can see here in light blue, and what we fitted is the black part. And we basically just flatten and cut it off, the sine wave. And the sine wave just has a parameter for the frequency, the amplitude, and how much it is kind of like baseline shifted. So that's relatively simple. And the reason we did that is because with that in hand, we can begin to understand what might happen if we have more than one event. So if you consider a pair of 2 events now, a first event in red and a second event in blue, and there's some time temporal offset between the 2 events, then you get a scenario that is shown here. And based on thinking of these sort of activations as part of the sine wave, you can easily calculate what the difference between the 2 events looks like. So this is the activity of the first event minus the second event. And you will get that curve that is shown here. And you will get a formula for that curve that tells you how that difference will change as a function of how far apart in time the 2 events occurred. And the formula looks a bit complicated, but I guess the basic understanding is that we have one prediction about the amplitude that is scaled by delta, not surprisingly. events further apart will elicit larger difference curves, and a prediction about the frequency of this difference sine wave in itself as a function of that delta. And we can calculate that in some pretty easy way. We also get some very basic predictions that are more or less obvious, which is that during the first part, the difference between the first and second event will be positive because the first event will dominate the second event. And then once they start falling down the curves, the second event will be higher and the order ring will be reversed. So what's important to keep in mind is that this example of a pair of two events generalizes to multiple events if you just use a different metric. So here we just looked at the difference between the first event and the second event. You could also take a regression at every point in time. If you imagine you had several events here, and we at every point in time we basically quantify to what extent are the events ordered in the way that we assume they should be ordered, with the first event having the highest probability, the second event having the second highest probability, the third event the third highest probability, and so forth. So we can quantify that by a regression, and then this regression coefficient will have the same properties that we have sort of shown here for the difference between the two events. And I'll show you in a minute that that's true empirically, at least. Okay, so now let's look at the sequence trials. As I said, each sequence trial involved all the 5 different image categories. We showed them in slow speeds of 2 seconds between the images up to fast speeds of 32 milliseconds between the images. And when we now look at the time courses of the classifier probabilities for each of the different classes, then we can see that this works really beautifully well for the slow sequences. So if we have 2 seconds between the images, the probability of the first class goes up and then the second class and so forth, and we can easily infer which order things happened in. Of course, the ordering of which class was actually first and which was last was counterbalanced across trials, was randomized across trials and between participants too. So when we go a bit faster to 512 milliseconds between images, then we can see that it gets messier, but it's also actually clearly visible by eye. The red stuff is in the beginning, blue stuff is in the end, and the green and orange ones are in the middle. And you can infer the order even by eye. And now if we go into these temporal regimes in which it gets really tricky, and that might be more interesting for studying replay of only having a few tens of milliseconds between the events, then you can see everything is really flat and the classifier is really struggling here. But you can still observe that there is the basic patterns that we have seen before are retained. So in the 128 millisecond condition, as in the 32 millisecond condition, you see the red classifier probability time course, which reflects whatever category was first, comes up first, and the blue one comes up last, and the other ones are in between. So to kind of not be only like thrown back to our visual impressions, we can quantify that statistically by doing what I mentioned before, fitting a regression slope for every TR that quantifies to which extent these probability time courses are in the order expected. So when we do that, we get a pretty clear picture that corresponds to our predictions. So the yellow line now is the condition in which the sequences were shown relatively slowly. And you will see that the regression slope initially goes up, which indicates forward orders, meaning the first element has the highest probability, the second one the second highest, and so forth. And then after a while it falls down. And that's very similar to the prediction I had shown you in the beginning. And the same overall pattern is true for all speed conditions. Of course, the amplitudes get lower and the frequencies of this sort of wavy thing here change. But the overall pattern is retained. And if we just sort of look at the forward periods when it's peaking up here and the backwards periods when it's peaking up here, and we're not taking just the periods that we observe from the data, but the periods that we have predicted based on the single response time courses that I showed you initially, then we get significant effects even in a 32 millisecond condition. And the effect sizes aren't particularly small compared to being used to fMRI. And how fast is this? We can get statistical evidence that this, like at least downwards bowing of the curve is beyond chance. So here is, to be more precisely, the prediction that we had made just on this very basic insight about what happens when you have two events and we look at the difference between the two events, that we get this interference curve. And when we plug in the actual numbers for delta from our task, this is the prediction that we get. And you can see it really corresponds quite nicely to our observations in the task. With the more temporal distance between events, we have sort of a larger amplitude and slower frequency response in the regression coefficients. We can, of course, correlate the model predictions directly against the observed ones, and we find reasonably high correlations throughout. So this effect is not limited to this regression approach that I mentioned before, but it also will lead eventually to the sequential progression through patterns, the pattern transitions that we had observed before, as I mentioned. So what I'm plotting here is the average event position of the maximum decoded event as a function of time. And as you can see, it again has the same shape, only now sign flipped. So for the, sorry about that. For the slowest condition with 2 seconds between the images, you see that the earliest events are predicted earlier. So it tends to predict events of position 1 or 2 or 3 here, and then later it predicts events of some later serial position. And that general pattern holds across all speed conditions, and again is kind of significant even in the fastest cases. Okay, so that seems nice, but of course it's like a simplified experimental case that is not going to be what will happen when we try and understand replay. And one of the features of studying replay is that we usually have resting periods, extended resting periods, and we don't know when exactly replay occurs within these resting periods. So we started thinking about what are the statistics that we can derive from these insights that we could apply to an entire resting period. And one interesting sort of approach is to look at the frequency spectrum of that regression coefficient. As I showed you before, the regression coefficient, if there was a fast sequence or a medium fast sequence, is going to start to wiggle in a specific frequency. And we can make predictions about that. So to kind of really illustrate that fact, if we were to take all like 15 sequence trials from one participant and just concatenate the data. So that corresponds to a participant lying in the scanner and thinking with about a speed of 2 seconds per item over and over again about the same sequence of images. Then what the data, the rest data would look like is what you see here on the bottom, like the yellow wiggling line, and it has a very clear particular frequency to it. If you do the same thing for the fast sequence events, then this is what it's going to look like, harder to see by eye, of course, and we can compare that to the rest period. So when we now do that systematically and we do a frequency spectrum analysis on the regression coefficients of these sort of concatenated time courses, then we see that they differ very systematically in the frequency spectrum. So the light gray line here is the rest condition, and that has a more or less typical 1/f fall off. And the yellow line is the slowest condition with 2 seconds in between, and you can see that it has a very clear peak in a particular frequency range. And interestingly, as I said before, we have this very, very simplified model that makes a ton of simplifying assumptions, but it gives us some ballpark measure about what is the frequency that you would expect that measure to the regression coefficient to start fluctuating in. And that prediction is shown with this line here, and as you can see, it's really not far off. It's pretty indicative of what we see when we look at slow sequences. And the same is true for the faster sequences, where the effects are, of course, much smaller, but we still see that the period where the experimental data and the rest data and the slow data start to differ most is exactly where you would expect it to differ. So now we continue to make our life harder to get closer to sort of the true challenges of an experimental setup, as we will face it when we will begin studying replay. And what we did is two things. One is we account for the fact that, as I mentioned before, during rest, you will not have one replay event, another replay event, and so forth, but you will record 5 minutes of rest in humans, and there will be 2 replay events, maybe, or maybe 5, or maybe 6, but we don't know. They will occur at random time points. The other sort of concession that we wanted to make is that, well, we have sort of optimized conditions in terms of the SNR that we have because our classifier works so well with over 65% accuracy. And that might not be true for hippocampal classifier in particular. So what we did is that we took the rest condition and then we inserted at random periods in time sequence events, and we varied how many of them we inserted, and we added noise to those sequence events to varying mixing degrees to basically see how much can the SNR be reduced and how few replay events can we have within a 5-minute period in order to be still able to detect the presence of these events statistically. So the first observation that we made then is that there is a very general basic property of the classifier time courses that is simply different between any kinds of events in which no reactivation happened and events in which reactivations happened. And that is the standard deviation of the probabilities over the 5 different classes. So maybe if you think back to the time courses of the probabilities of the 5 different classes, I can maybe show it to you actually. These ones are the ones that I'm talking about. And what you can see here is that when we have a lot of activations of the different image categories, the classifier probabilities, they really get squished together. So the standard deviation of them is relatively low. And that is an indication not of chance, but that's an indication of there's actually lots of reactivation of things that the classifier was trained on, and it just isn't sure which one it is because they all are temporally so close together. So that is this reduction in the standard deviation of the classifier time courses that we observe, and that sort of scales with the number of inserted sequence events. That's color-coded here from red to blue, and the red line, to be sure, is sort of a condition which the noise is maximum, so you wouldn't expect any effect there. But what's more interesting, and maybe the most stunning finding here, is that we can also look at the frequency spectrum and see if we can pick up these sort of artificially inserted lower SNR events there. And that indeed seems to be the case. So here you see sort of normalized frequency spectra. This is the case in which the inserted events were fast, and we only inserted 2 of them. And this is the frequency range that we would expect for the faster frequencies. And you can see that the higher the SNR, which is color-coded here, the larger we get sort of a bump in that specific frequency spectrum. You can also see that we get issues with harmonics that we haven't solved yet. Maybe someone who's more familiar with these techniques can tell me how to suppress those harmonic activations. But it's clearly in the right ballpark, and it gets better if we have more sequence events. So more sequence events is of course good, and higher SNR is also good. But importantly, even if we sort of more than half the SNR, quarter it, and we only have a few events, we might get a shot at seeing these sequences in the resting data. And distinguishing which speed they occurred in. So finally, and this is very preliminary data, as you can see from the resolution of the image, is that we, during the review process, we were actually asked by a reviewer to compare resting state conditions that we had recorded before participants were exposed to the task to a resting state period that we had recorded to after participants have been exposed to this task. And to kind of make that clear, we didn't optimize the task to like elicit any replay. It seemed possible that like any processing any kind of sequential activation might elicit that, but there was no mnemonic component or anything on it. But what we start to observe, and we still need to do some work to clarify this is really true, is that when we look particularly at the sequences that occurred in the session before the rest was done, then we find a bump in the frequency spectrum that indicates a relatively fast reactivation. So the gray line is the resting period before they have been exposed to the task, and the black line is the resting period after they have been exposed to the task. And this analysis is assuming that one of the 15 different sequences that happen in that particular session might have been replayed and is averaging over these 15 different possibilities. And as you can see, comparing the light gray line to the black line, we see in this faster frequency spectrum an indication that there might actually be reactivation in visual cortex following our task, which would be a quite nice additional proof that our method works. Okay, I'm realizing that I have been relatively fast to rush through, but maybe that's good that we have more time to discuss. So the quick summary that I wanted to give you is that we can use fMRI to investigate fast sequence activations. We, of course, need enough SNR and power for that. So the training set for the classifier will be really crucial. This regression slope approach that I explained to you just now seems to be somewhat more sensitive than the pattern transitions that we had used earlier in the science paper, but both of them should work in principle. And hopefully I can soon present you some studies in which we investigate actual replay and not just some methodological foundations. But you can look at our science paper from 2019 to already get a sense of what that is going to look like. So with that, I want to thank my lab. This is Leonard Wittkuhn that I want to point out, who's been spearheading all the sort of methods work that I talked about. As I mentioned before, if you're interested, you should go and read our paper from last year about sequential reactivation of non-spatial task states in the hippocampus. This preprint about the work that I presented now is out on bioRxiv and hopefully will be published at some point this year. And I also wanted to briefly mention that we will soon advertise for a new postdoc position. So if you're potentially interested, then you can reach out now and I can give you the details. All right, so with that, I think I'm happy to take your questions.
Thank you, Nico. I will clap on behalf of everyone. If you have questions, you can write those in and ask a question facility. Maybe I'll just kick things off. So perhaps you were saying this with your point about harmonics. I'm not sure I quite followed it. But when you're adding the little snippets of replay onto the background, are you somehow smoothing the edges, or could it show up as sort of high frequency edges that somehow making the task easier even in that setting?
Yes, we are not smoothing the edges. We have seen some artifacts, but the main comparison then will be down to the fast versus slow inserts. So because the edges should be present in both cases, they should actually be a bit stronger if we insert slow sequences versus fast sequences. And you can see the differences between the slow sequences, which are here, and the fast sequences, which are here. So you're right that ideally you would want to compare these two rather than just want to compare. Yes, does that make sense?
Yeah, no, that does make sense. Any questions from the audience? All right. Would you like to ask that question on the screen, Ben? I'll invite you up and let you do it if you want to. Oh, please ask for me. Okay. So we have a question from Ben asking, what would replay look like in cortex? Do you mean in cortex as compared to hippocampus, or, well, I'll let you interpret that as you will, Nico.
Yeah. Well, I guess that's part of the question that we're up to. So from the measurement side of things, setting aside some differences in the href, which I think the classifiers are reasonably well equipped to kind of generalize over, the measurement wouldn't make a huge difference. So that's one of the benefits of fMRI, that we have roughly equal sensitivity for cortex and subcortical areas, which is not necessarily true for MEC. In terms of what the replay content is about in cortex versus hippocampus, I think that is exactly the interesting question. So it's not necessarily true that we find play cells in the cortex. So the representational nature of the cells, even for single events, is different, and therefore the nature of the sequences among them are different. And I think that will be exactly the kinds of questions that we can answer with looking at replay at multiple sites at once, to understand how different aspects of the same task, for instance, the spatial location, but also the serial position within the task, or maybe some attentional state or sequences of attentional states, are reactivated in parallel and in a coordinated fashion across cortical sites. Sure, happy to.
Anyone else? Well, while we wait, I guess I'm also curious how you see this, or sort of what your perspective is on sort of sleep and replay, which I guess is not something necessarily that you can do with this method, but how you separate out these different types of replay that could be occurring.
Yeah, so I think this paper that I mentioned from Freya is going to be really interesting. She didn't look necessarily at sleep in that paper, but the general understanding so far was that the replay that happens during sleep is more related to consolidation, and the replay that happens on task might, in some instances, more be related to planning. And if that is true, that makes specific predictions about the coordination aspect of it too. Because for coordination, you would really have to have in-sync activation of hippocampal sequences with particular cortical sites. You could maybe even then make predictions about where you can decode particular memories from afterwards, once they have been consolidated into those memories, if you had access to that kind of information. And I think that's particularly interesting. And the replay on the task then might be completely different in nature, because it refers more to a planning function or maybe to a generalization function. And that, again, might be reflected to sort of co-activation with different areas as opposed to sleep. Yes, I can actually imagine that even during the on-task periods, we will see more completely hippocampus independent replay events. So similar to the Schaefer paper, that certain areas that are involved in processing the task and understanding aspects of the task will independently of the hippocampus replay the particular sort of aspect of the task that they are involved in representing. And they will do that completely independently of the hippocampus to learn sort of structure. Whereas during sleep, it might be really the case that the hippocampus is kind of the pacemaker and is starting off the whole process to begin with.
All right, we have a question from Ben Hong. When replay happens in the rodent hippocampus, the sequences can reoccur in different orders for replay and preplay. Would it be possible to detect this difference in fMRI using your technique?
That is a good question. So if you refer to the different orders for replay and preplay, You mean the forward versus backward ordering? That is a good question. And it's a problem for us that I'm happy to admit freely, because if I go back to my slides, you can see that this metric that we have is kind of symmetric in a way that it has a forward order and then it has a backward order. So it's kind of hard to tell if you see like just this part of the curve, then it could have been the backward part of the forward replay, or it could be the forward part of the backward replay, if you understand what I mean. And the way to solve this is, of course, to look specifically in your data, not only for fluctuations in a specific frequency range, but for the ordering of like the upwards bump and the downwards bump. And we haven't done that to what extent that's possible under realistic conditions. But I think there's a handle on it, but it's not easy because of the particular nature of the fMRI signal. That will make that a bit harder.
I guess more generally, is there a way to use these similar methods for going after phenomena that aren't necessarily replay as we traditionally construe it, but you're just showing ways that you can get much higher temporal resolution in certain instances from fMRI?
Yeah, I think so. Definitely. I mean, there might be other sort of areas. I mean, that's maybe then comes down to the question: what do we want to call replay, and what do we not want to call replay? Is that necessarily sort of any sort of fast temporal like sequence? Is that replay? For instance, there are
exactly. I mean, like, oh yeah, just thinking in motor cortex, right? If you see unfolding sequences, it may not be interpreted as replay, but
exactly. So there's temporal pattern completion. I think people have shown that in visual cortex. If you have a well-trained visual sequence and you start flashing the first two elements of it, then the rest of it gets auto-completed. And I would definitely imagine this is something that happens when you have like a well-trained motor sequence, that when the first two things are initiated, the dynamics will kick off and the rest of them will be also initiated regardless of the movement. And for that, I think that we can definitely use the same tools. They might be potentially easier to use because it's easier to record motor and visual activation than it is to record hippocampal or like decode hippocampal activation.
All right, we have a question from Yaron He. I was wondering how valid you think it is to apply the similar classifier to hippocampal data. As you mentioned, the accuracy you got in the 2020 paper on visual cortex compared to the previous paper with NIV in hippocampus, the accuracy was at 11%. The visual cortical classifier seems to be much more accurate. For my own data, I found a very low multivoxel variation of the hippocampal data compared to visual cortex data, and the classifier didn't work very well.
Yeah. So that relates to these SNR manipulations that we have done here, sort of as an attempt to kind of account for that. So the SNR means basically that's the mixing proportion of noise to the signal. So we insert these artificial sequences, but then an SNR of 1/2, we just instantiate it as, okay, sort of that's half of the rest data, that's just noise plus half of the sequence data. That should roughly lead to half of the decoding accuracy, probably less, if I would have to take a guess. And we definitely see that the SNR is a bottleneck. If it gets really low, you can observe that here with 6 sequence events across a 5-minute period, then the effects get relatively small. So I guess you need to have maximum SNR. But it's not true that if you had like only 1/8 of the SNR, that you cannot see anything at all anymore. It's just kind of a graded function. It seems to degrade relatively gracefully, not very catastrophically if you have enough sequence events. But it's also kind of referring to a more complex function. We also, in this data set, we try to decode from the hippocampus, and we cannot decode anything from the hippocampus during the task. but there have been some studies that indicated that even tasks that do not seem to be hippocampal dependent during task processing, so do not elicit any hippocampal fMRI activity, seem to be dependent on the hippocampus for consolidation. So in these cases, we will be stuck with like lacking a training set basically for the hippocampus. But the same is true for the animal work. So in our work and the work that you mentioned, we might be stuck with only studying visual activations because the task is not interesting enough for the hippocampus. But it doesn't necessarily mean that there's no hippocampal reactivation during rest, only that we don't know how to interpret the data.
Thank you. Any other questions? All right. Well, thank you for your time, Nico. I think this is a really fascinating, promising technique, and can't wait to see. Actually, here's the final question. What is your, do you have a target for first study you want to do with these methods?
Good question. I am interested in sort of the occurrence of task-sensitive information in the hippocampus and outside of the hippocampus. So we have observed in a bunch of previous studies in the orbitofrontal cortex that the orbitofrontal cortex is involved in, over time, understanding which aspects of the task are and which one are not. And we have seen in the 2019 paper that to some extent the hippocampus is already reflecting that. But what I'm really interested in the future is understanding whether the hippocampal replay has any role in that learning process of understanding which aspects of the task are actually relevant to begin with. Or if the replay, I mean, in the very early instantiations, the replay is really dumb, quote unquote, because it's episodic, meaning it would reflect the entirety of the event as it was perceived, rather than the filtered event only reflecting the relevant aspects that are necessary to understand structure. And I think I'm interested in understanding that. For instance, how does the content of replay change over time as people understand what the episodes actually mean?
Fascinating. All right. Well, thank you for your time. And let me also just close the note about this series. So we're now going to pause for the summer, and we'll start up in the next term. This is the last talk in this series until the next term starts up. So thank you very much again. See you, everyone.
Thanks, everyone. Bye-bye. Thanks, Andrew.